Hello everyone, my name is Snehlata and I'm a PhD student and a PMRF fellow in the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today we will continue with the next lecture of the chapter Photosynthesis in Higher Plants. So till now we have studied about the light reaction and a part of dark reaction, that is Kelvin cycle. And we saw that the Kelvin cycle is the universal cycle. Now, the question was how it is universal cycle if there are multiple number of other cycles also present. Okay. Then we also studied about C4 plants in which predominantly C4 cycle takes place, and we saw that what are the cells that participate in that C4 or what are the major cells of photosynthesis present in C4 plants. Now, in today's lecture, we will start with the discussion about the advantages and disadvantages of C4 plants. So, if we talk about the advantages of the C4 plants, so one of the major advantages, if you remember, we said that C4 plants are the most productive plants. So, in terms of their production, they are most productive. So, C4 plants are most productive as compared to any other plant type. Okay. Now, these plants have tolerance to a wide variety of stress. Okay. So they can tolerate low carbon dioxide concentration, low carbon dioxide concentration. Okay. It means they can sustain CO2 stress. So they can sustain CO2 stress, right? Now, apart from this, they can also tolerate high temperature. So they can tolerate high temperature. So it means they are, you can say, not temperature resistant, but can sustain the temperature stress. Then, if they can sustain high temperature, with this, you can also remember that they can also tolerate high light intensity. So they can also tolerate high light intensity. Okay. Then they can also tolerate water stress. It means the low water concentration. So they can tolerate water stress, which means that if these plants do not have enough water, then also they can live. Okay. So it means they can sustain in low water, less or low water concentration. Now, why does this happen, or how do they sustain in the low water concentration? This is because of the presence of a special kind of stomata. Okay. So the stomata, we know this is used, they undergo opening and closing and they prevent water loss, right? So the stomata that is present in the case of C4 plants, these have sunken stomata. So these C4 plants have sunken stomata. Okay. It has sunken stomata and hence it prevent water loss. So even if these plants are in low water condition, they can sustain or they can survive there. Okay. Now, apart from this, it can also tolerate, these plants can also tolerate high salinity stress. High salinity stress. By salinity, what I mean? It means salt concentration. So salinity means salt concentration. So these can also sustain high salt concentration. Okay. And this might be the reason that these plants are most productive, because even if the concentration of carbon dioxide is low or water is low, they are efficient to survive and hence can be most productive. Now, this is about the advantages. But what are the disadvantages of the C4 plants? So we do not have much of the disadvantages of C4 plants. The only disadvantages, so if we talk about a disadvantage, so we do not have much. If we talk about this, so we have only one, that is, it is sensitive to low temperature. Okay, so it is sensitive to low temperature. And this is because it has an enzyme. The C4 plants have enzyme known as pyruvate dikinase. Enzyme known as pyruvate dikinase. And this enzyme, pyruvate dikinase, that plays a role in the photosynthesis. Okay. And this pyruvate dikinase, it is sensitive to low temperature. So it is cold sensitive. So under low temperature condition, it means under low temperature condition, it will not work. So in low temperature, it will not work, or you can say its efficiency will reduce. So this is the only disadvantage, or one of the, you can say, major disadvantage of this plant, that it is temperature sensitive. Okay. So now we have discussed about what are the C4 plants, what are the major cells that involved in the photosynthesis of the C4 plants that we discussed in the last lecture. And now we have seen the advantages and disadvantages of the C4 plants. Now we know that C4 plants are named because of a dark reaction that takes place in them. And this dark reaction is known as C4 cycle. Right. So what is C4 cycle? So C4 cycle, it was given by Hatch and Slack. Okay. So C4 cycle, this cycle is given by scientist named Hatch and Slack. Okay. And because of this reason, this C4 cycle is also known as, Hatch and Slack pathway. Also known as Hatch and Slack pathway. Okay. Now, in the C4 cycle, okay, so another advantage of C4 cycle that is most important is it does not undergo photorespiration. Does not undergo photorespiration. And we will see why this happened. Okay. So this C4 cycle we're talking, so it was given by Hatch and Slack, and it is also known as Hatch and Slack pathway. Okay. Now, in C4 cycle, it occurs when riboscope, we know enzyme riboscope that we have studied when we were studying about the C3 cycle, and we saw that the riboscope is bifunctional, that it can act as both carboxylase and oxygenase, right? But in the case of C4 cycle, this riboscope acts only as carboxylase. Only as carboxylase. Okay. Now, why this happened? We'll see. So it means its oxygenase property, it does not mean that it cannot bind to oxygen, but somehow it does not get sufficient oxygen to bind, and hence it cannot act as oxygenase in the case of C4 cycle. It means that it will only bind to CO2 in case of C4 cycle. Okay. Now, in the case of C4 cycle, the fixation of carbon dioxide, because we are studying about dark reaction, so here what happens? Carbon dioxide undergo fixation, right? So in case of C4 cycle, fixation of carbon dioxide takes place. It takes place in two cells. And these cells are what? These are mesophyll cells. These are mesophyll cells and bundle sheet cells. sheath cells. And we saw what is the difference between mesophyll cells in the case of and bundle sheath cells in the case of C4 plants or during C4 cycle. Okay. So we will come to the steps. First, let's draw the cycle for this C4. So, like I said, that this cycle takes place in two types of cells, right? So let's draw. You have one type of cell, right? And then we have another type of cell. Okay. So the first cell is this is the mesophyll cells. This is mesophyll cells. Now, what happens in the case of mesophyll cells? So, what is difference between the mesophyll cells and bundle sheath cells in the case of C4 plants? In mesophyll cells, it does not have ribisco, right? So, no ribisco. Unlike C3 plants, in which the mesophyll cells have ribisco, but in C4, it does not have ribisco, but it has another enzyme known as peptidase. Okay. Now, this peptidase is involved in initial carbon dioxide fixation. So, what happens from the environment? The CO2 comes inside the cell, right? Here, this carbon dioxide, in the presence of a substrate known as phosphoenol pyruvate, known as phosphoenol pyruvate, or you can also say it as PEP. And this PEP is what? 3-carbon molecule. So it comprises of three carbons, right? So this carbon dioxide binds to phosphoenol pyruvate in the presence of the enzyme that is known as peptidase. Peptidase, which means PEP carboxylase. PEP carboxylase. Okay. So it binds to it and gets converted into, because this phosphoenol pyruvate, it is of 3-carbon, right? And this carbon dioxide, this is 1 carbon. So it will gets converted into a 4-carbon molecule known as oxaloacetic acid. So it gets converted into oxaloacetic acid. And this oxaloacetic acid is 4-carbon, right? Now, this oxaloacetic acid, it's by utilizing what? By utilizing NADPH, NADPH, it's get converted into NADP plus. Okay. And by utilizing this NADPH, it gets converted into malic acid or aspartic acid. Okay. So it gets converted into malic acid or aspartic acid. Now, this aspartic acid from the mesophyll cells, it comes to the bundle sheath cells. It comes to the bundle sheath cells. Okay. So this malic acids come to bundle sheath cells. And this is also 4-carbon, right? Okay. So what happens now? This malic acid again gets converted into, here it comes to bundle sheath cells, and it gets converted into oxaloacetic acid. Oxaloacetic acid that I'm representing by OAA. Okay, so oxaloacetic acid can also be represented by OAA. And here, what happens? Here, like we saw in the case of mesophyll cells, when oxaloacetic acid was converted into the malic acid, the NADPH was converting into NADP plus, right? Now this reaction is getting reversed. So here, the NADP plus, it gets converted into NADPH. Okay. And this oxaloacetic acid then gets converted into pyruvate. And when it gets converted into pyruvate, it releases carbon dioxide. Okay. So here, the carbon dioxide that was taken by the mesophyll cells, it gets converted or it gets released from oxaloacetic acid in the bundle sheath cells. Okay. And this carbon dioxide, because we know the bundle sheet cells, they also consist of chloroplast, right? So the bundle sheet cells are the chloroplast. This is a chloroplast. And in the chloroplast, it has what? This granule thylakoids, right? So it's not actually granule. We have thylakoids here, right? And these thylakoids had what? We saw the difference between the chloroplast that is present in the bundle sheath cells and that is present in the mesophyll cells. So in the case of bundle sheath cells, this chloroplast will have only PS1, photosystem 1, right? Here. So this CO2, it enters into the carbon this chloroplast, and here what happens? So this is the chloroplast, and here C3 cycle takes place. Okay, here C3 cycle takes place. And this is the reason why we call this C3 cycle as universal cycle. Because what we saw, although the process of initial fixation of carbon dioxide is different, but ultimately it comes to the chloroplast where the C3 cycle takes place for the final carbon dioxide fixation. Okay, so in this chloroplast, the CO2 enters. And the second thing, so if you remember the C3 cycle, it involved the use of both NADPH as well as ATP, right? Now, from where the NADPH comes from? So the NADPH, we can see here, this NADPH that is formed, it also goes to the chloroplast and then it is used for the C3 cycle reaction to take place. Okay. Okay, so here, and these chloroplasts, they have only PS1. Only PS1, because we know the PS1 are present on the exposed region while the PS2 are present on the non-exposed region. But this chloroplast in the bundle sheet cell, they do not have any unexposed region. So only the PS1 is present. Okay. And what happens to this pyruvate? So this pyruvate then it gets used or for the regeneration of phosphoenol pyruvate. Okay, so it undergo regeneration. Okay, so this is what happens in the C4 cycle. So in summary, if you see this carbon dioxide, it is entering into the mesophyll cells, right? And this, if you remember the cell wall of this mesophyll cells these are thin cell walls, right? These are thin cell walls, while the bundle sheath cells, these are thick cell walls. So once the carbon dioxide enter here, this carbon dioxide is not allowed to escape. So it cannot escape, and hence the concentration of the carbon dioxide inside this bundle sheath cells, it increases. Okay. So what happens? The carbon dioxide is taken by phosphoenol pyruvate in the presence of the enzyme known as peptase, and it gets converted into 4-carbon molecule, that is oxaloacetate. Oxaloacetate further gets converted into malic acid, and this malic acid is then transported. It is then transported from this mesophyll cells to bundle sheath cells. And in bundle sheath cells, it ultimately goes to the C3 cycle in the chloroplast. So here the C3 cycle takes place. Okay. So if you talk about the steps, so what are the major steps that are involved here is, let's see. So the first step that is involved is carboxylation. The first step is carboxylation. So what happens in the carboxylation? Carboxylation, we can see phosphoenol pyruvate undergo carboxylation by the addition of this carbon dioxide, right? So here, phosphoenol pyruvate, that I am writing as PEP, undergo carboxylation in the presence of peptase, but in the presence of peptase, right? Now, this is the first step. Now, what is the second step? After the carboxylation, we saw that the oxaloacetic acid is formed, that gets converted into malic acid. And then what happens? Transportation, right? Then what happens is transportation. And what happens in the transportation? This C4 acid, like the 4-carbon containing acid, here because the what is getting formed is the acid, right? The malic acid or aspartic acid or oxaloacetic acid. So this four carbon molecule acids, the four carbon molecule acids are then transported are transported from mesophyll cells, right? From mesophyll cells to bundle sheath cells to bundle sheath cells. Okay. So this is the transportation. Now, what is the third step? Third step is decarboxylation. Because in the first step was the carboxylation, the third step is decarboxylation. So, what kind of decarboxylation is happening here? So if you see this step, this is your third step. Here, this oxaloacetic acid is undergoing decarboxylation to form pyruvate. Okay. So in decarboxylation, carbon dioxide is removed from oxaloacetic acid. It is removed from oxaloacetic acid to form pyruvate. Okay, to form pyruvate. Now, what is the fourth step? The fourth step is transformation. Transformation. Now, what happens in the transformation? C3 acid is formed, right? So this pyruvate is also known as, this is also known as pyruvic acid. Pyruvate is also known as pyruvic acid. So what happens? Transformation. So from the four carbon molecule, 3 carbon molecule is formed. So from 4 carbon acid, 3 carbon acid is formed. 3 carbon acid is formed. Okay. And then finally is the regeneration. Finally is the regeneration. So what happens in the regeneration? This 3 carbon molecule acids, the 3 carbon molecule acids undergo conversion to form PEP. So 3 carbon acids undergo conversion to form what? To form PEP or phosphoenol pyruvate. Okay. So these are the steps that are involved in the C4 cycles. Now, what observations do we have from the C4 cycles? So if we see, the important point is in C4 plants, because C4 cycle we know it takes place in C4 plants, right? But in C4 plants, what you can say, not only C4 cycle, but C3 cycle also takes place, right? So in C4 plants, both C4 and C3 cycle takes place. Okay. Okay. Now, we have seen the C4 cycle. Now, what are the energy expenditure that takes place in the C4 cycle? So, if you talk about the C4 cycle, it comprises of two things, or you can, for more clarity, you can write here C4 plants. Okay, energy expenditure in C4 plant during dark reaction. During dark reaction. Okay, so if we talk about the one carbon dioxide fixation here, if we talk about one CO2 fixation, so in this case, let's see how much ATP and how much NADPH is required. So because it is C4 plant, so we have both C3 cycle and C4 cycle. Now we remember, or we have seen in the previous lecture, that in C3 cycle, how many ATP and how many NADPH is required for one carbon dioxide fixation. So for one carbon dioxide fixation, we require 3 ATP. We require 3 ATP molecule, 2 during reduction and one during regeneration. Regeneration, right? So we have three ATP molecules and two NADPH. 2 NADPH, and that is required during the reduction process of C3 cycle. Okay. So this is for one carbon dioxide fixation. While in the case of C4 cycle, we need 2 ATP. We need 2 ATP, and here we are seeing that NADPH is getting used in here, right? It is getting used. But if you come across and see in the bundle sheet itself, this NADPH is again getting regenerated, right? So if you talk about the net NADPH that is required in the C4 cycle, it's 0. So because one is getting used up and then this one is getting formed, so the net NADPH will be 0. So we need 2 ATP and no NADPH, you can see. Okay. So this is about one carbon dioxide fixation. Now if it talks about one glucose production, so if you need one glucose production, then how many in the case of C3 cycle and in the case of C4 cycle, how many ATP and NADPH will be required? So if you see, because for one glucose, you will need 6 carbon dioxide fixation, 6 molecule of carbon dioxide fixation. So it's simple. So it will require 18 ATP molecule, 18 ATP molecule, and 12 NADPH, right? And out of 18, 12 will be from reduction. So 12 ATP for reduction and rest will be for regeneration, right? Now, in the case of C4 molecules, because one carbon dioxide fixation requires 2 ATP, so 6 carbon dioxide fixation, it will require 12 ATP, right? So it requires 12 ATP molecules. Okay. So if someone asks that in the case of C4 plants during a dark reaction, how many ATP, a total of how many ATP and how many NADPH are required? So total in the case of C4 cycle, dark reaction, you will have 18 plus 12, 30, 18 plus 12 ATP, right? Including both C3 and C4, that is 30 ATP and 12 NADPH. So this is the total energy expenditure in the C4 plant during dark reaction. Okay. So this is about the energy expenditure. Now, we said that the C4 cycle, it does not undergo photorespiration, right? So what is the reason that it does not undergo photorespiration? So what we are seeing here, the first step, if you look into the cycle, the first step is the initial carbon dioxide fixation. Where it is taking place? It is taking place in the mesophyll cells, right? And in the case of C4 plants, these mesophyll cells, they do not have rubisco. So the first reason is initial CO2 fixation. The initial CO2 fixation takes place in mesophyll cells, right? In mesophyll cells. And these mesophyll cells in C4 plants, they do not have rubisco. Do not have rubisco. So if they do not have rubisco, in this case, what happens? The carbon dioxide, because this their PEPK is not rubisco, so it cannot act as an oxygenator, and hence photorespiration will not take place, right? Now, the second thing is, what happens in the case, now this carbon dioxide that is taken up by the mesophyll cells, it, by undergoing several transformations, finally reaches to the bundle sheet cells. And we know that in bundle sheet cells, this chloroplast, it consists of rubisco. It has Rubisco. So the question is, the Rubisco can bind to the oxygen here, right? But if you see this chloroplast, these chloroplasts have the grana, sorry, they have the thylakoid that they contain only PS1, right? The thylakoids contain only PS1. So if you see that PS1, if you remember the cyclic photophosphorylation, what happened in that case? In that case, we know that there is no oxygen in evolution or formation takes place when only PS1 is active, right? So you can write that the C3 cycle of C4 plants takes place in bundle sheath cells, right? It takes place in bundle sheath cells. And these bundle sheath cells, the chloroplast, have only PS1, And the PS1 we know it does not involve in water splitting. Okay. PS1 is not associated with water splitting, right? And we know that oxygen evolution takes place only when the water splits, right? Because we know the reaction here, the water, it undergoes splitting to form H plus electrons and O2, right? So this reaction takes place only when water splitting takes place. So if there is no water splitting, it means no formation of oxygen. So although this bundle sheath cells, they have Rubisco, but because there is no oxygen formation, so the Rubisco cannot bind to the oxygen, right? So this is the second case. And the third point is, we know that the cell wall of this bundle sheath cells, they are thick. Okay, they are thick and impermeable. So the carbon dioxide that gets released in the bundle sheath cells, they cannot escape, and hence it causes the increase in the concentration of the CO2 in the bundle sheath cells. And we know that the Rubisco have high affinity, right? So we know. So what we can write, if we bring together all the points, so what we can write is that Rubisco have high affinity for carbon dioxide, right? Have high affinity for carbon dioxide. And this bundle sheath cells have thick cell wall. So these cell wall, they do not allow, so these cell wall does not allow carbon dioxide to escape. Now, the carbon dioxide will not escape. It will cause the increase in the concentration of carbon dioxide, right? So it will lead to the increase in the concentration of carbon dioxide, and ultimately it is only the carbon dioxide that will bind to Rubisco. And if carbon dioxide bind, the photorespiration does not take place. Okay. So in summary, what we can write is, that C4 plants have a mechanism to increase the carbon dioxide concentration in the cell. To increase carbon dioxide concentration in the cells. And if this concentration of carbon dioxide will increase, what will happen? Rubisco will only bind to carbon dioxide. So it will ultimately leads to binding of Rubisco to CO2. And because of these reasons, this C4 cycle does not undergo photorespiration. Okay. Okay. Now, so we have studied about the C3 cycle, we have studied about the C4 cycle. Now, and then C2 cycle, or you can say photorespiration. Now, the last cycle that we'll study is CAM cycle. So what is CAM cycle? The CAM cycle is known as cressolation acid metabolism cycle. So it is known as cressolation acid metabolism cycle. Okay. Now, where does this cycle takes place? So this cycle occurs in plants that are succulents. Okay. So this cycle occurs in succulents. Example, opuntia. Example is opuntia. Okay. Now, because these are succulents, so we know that they have thin cuticles and sunken stomata. One second, just a second. Okay. Yeah. So, this CAM cycle, it takes place in the succulents. So, for example, opuntia, and these succulents, they have thin cuticles and sunken stomata. Just a second. Okay. Sunken. Okay. So sunken stomata, this is used for what? To prevent water loss. Let me know. Okay. Now, these stomata, the first one is they are sunken. They are sunken. And the second is these stomata are photoactive. Okay. So these are not photoactive, means they are not active during the daytime, but they are active during the night time. Okay. So they have photoactive stomata, means they are active during night. Okay. And okay, so this is about the plant in which the CAM cycle takes place. Okay. Now, where does this cycle take place? So we saw that in the case of C3 plant, the cycle takes place in only one type of cell, that is mesophyll cell. While in the case of C4 plant, it takes place in the two type of cell. But in the case of CAM cycle, what happens? It takes place in single type of cell. So it takes place in single type of cell. Okay. Single type of cell, but at two times. Okay. But at two times during day and night time. But at two times. Okay. That is during day and night. Day and night. And because of this, it is known as, because here what is happening? There is difference in the time of the cycle to take place. So this is also known as temporal difference. So it means that CAM cycle takes place in temporal difference. Okay, difference. Okay. Now let's see what happens in the CAM cycle. So, like I said, it takes place during the day and during the night. So let's first see what happens during the night time. Okay. So during the night time in the mesophyll cells, and also it takes place in what? Single type of cell, that is mesophyll cell. That is mesophyll cell. Okay. So during the night time, what happens? We have our stomata. Okay. Now, So this carbon dioxide enters, and this carbon dioxide, again, it in the presence of PEP. Okay. So if we have PEP here, then the enzyme that will be here is PEPcase, right? So, and now we know this PEP is 3 carbon. So in the presence of carbon dioxide, it will get converted into 4 carbon molecule. And this 4 carbon molecule is oxaloacetic acid. So we have oxaloacetic acid, OAA, that is 4 carbon. Okay. Now we know that this 4 carbon, it will get converted into malic acid. Malic acid. Now this is happening during the night time. Now this malic acid, and this is again 4 carbon. This malic acid, what happens? It gets accumulated in vacuole. We know the plants have large vacuoles, right? So it gets accumulated in the vacuole. Okay. This malic acid gets accumulated in the vacuole. And because what happened here is this is the acid, and this acid is getting accumulated into the vacuole, then this process will be called as acidification. Okay. So it will be known as acidification. So this process, or you can, because it is happening during the night, it is also known as dark acidification. Okay. So here what is happening? Here, dark acidification is happening. Okay. And what is dark acidification? It is accumulation of malic acid in the vacuole during night time. So accumulation of malic acid in vacuole during night. Okay. So this happens during the night. The stomata opens, and because what I said that here the stomata is photoactive, so they get active in the night. So in the night the stomata opens, the carbon dioxide enters, and it binds to PEP, and then gets converted to oxaloacetic acid and malic acid. And this malic acid gets accumulated into the vacuole during the night time. Now what happens during the daytime? During the daytime, what happens? This vacuole, this accumulation of malic acid in the vacuole. So now during the daytime, this malic acid gets released from the vacuole. Okay. So during the day, we have vacuole, and this we have what? Malic acid accumulation. Now during the daytime, this malic acid get out of the vacuum, and then it gets converted into pyruvate. Okay. So it gets converted into pyruvate. And once it gets converted into pyruvate, we know pyruvate is what? It is 3 carbon molecule. Then that leads to the release of what? That leads to the release of carbon dioxide. And this carbon dioxide, then it goes to the chloroplast, right? It goes to the chloroplast. And in the chloroplast, what happens? In the chloroplast, C3 cycle takes place. Okay. So here also, although the name of the cycle is CAM cycle, but it leads ultimately end up in the C3 cycle in the chloroplast. And this pyruvate, what happens to the pyruvate? This pyruvate gets converted into PEP. Okay. It again gets converted into the PEP, and then the PEP is used for the next cycle. So this is what happens in the CAM cycle. The only, if you look at the steps that happen in the C4 cycle and in the CAM cycle, the steps are same. The intermediates that are formed are same. The only difference is in the case of CAM C4 cycle, these were happening in 2 different type of cells. Like, so they have spatial differentiation. While in the CAM cycle, these are happening in the same type of cell, but at 2 different time points. Okay. So this is the difference. And here, when the acid was getting accumulated in the vacuole, this was known as dark acidification. But when this acid got released here during the daytime, this process is known as deacidification. So here the acid is getting released, and this process is known as deacidification. Okay. Okay. So this is about the CAM cycles. So with this, we have completed all three type of, or you can say all four type of cycles. Now let's see what are the differences or comparison between C3, C4 and CAM cycle. Okay. So the first difference or feature on the basis of which we can differentiate is Kranz anatomy. Okay. So the first feature is Kranz anatomy. So we have seen the Kranz anatomy is what? It is crown-like structure, crown-like structure, right? And this structure was present in only a special type of plant, and that is what? C4 plants, right? So in C4 plant leaf, if you see, the Kranz anatomy is present, while this is absent in both C3 and CAM. Now the second feature is about photorespiration. Photorespiration. So the photorespiration, it takes place in C3, we know. Does it take place in C4? The answer is no. Will it take place in CAM? So if you look at the CAM cycle, will photorespiration takes place? The answer is no, right? Because here also there is a mechanism of increasing the concentration of CO2, right? And the initial enzyme that is involved in the fixation of carbon dioxide from the environment is what? PEP, right? Sorry, pepcase. And pepcase, it does not have oxygenase activity. It only have carboxylase activity. So the photorespiration will not take place in CAM as well. Okay. Now, if we talk about the morphology of chloroplast, so we saw a term called dimorphic chloroplast. Dimorphic chloroplast. It means that we know that we are this photosynthesis takes place in leaves, and in leaves, specifically in the mesophyll cell, and in the mesophyll cells itself, it takes place in the chloroplast, right? Now, the dimorphic chloroplast means that you have leaf, say you have leaf, this leaf has different cells, mesophyll cells, okay? So in one mesophyll cell, it has one type of chloroplast, while in another mesophyll cell, it has different type of chloroplast. Okay, so it has suppose here in one mesophyll cell, it is one type and different, second has second type of chloroplast. Then such type of chloroplasts are known as dimorphic chloroplast. Okay. So the dimorphic chloroplast we did not see in the case of C3. So in C3, there is only one type of chloroplast present, and that is what? That is granule chloroplast, right? Because it has this tag, grana, and they are connected like this. While in the case of C4 plants, we saw this dimorphic chloroplast because it has both. So yes, it has both granule and agranule chloroplast. So granule means the thylakoids will stack together, while agranule means the thylakoid will just scattered in the chloroplast. So it has both granule and agranule chloroplast. And the CAM has only one type of cell, that is mesophyll cells, and these mesophyll cells have granule chloroplast. So they do not have dimorphic chloroplast. Now the fourth is carboxylation. So in case of C3 plants, how many times carboxylation takes place? So if you remember, the carboxylation took place only once, right? When the carbon dioxide bind to RUBP, because in the case of C3 plant, the substrate was RUBP, right? So carboxylation, it takes place one time, only one time during one carbon dioxide fixation. While in the case of C4 plants, the carboxylation, it takes place twice, right? Two times. How? Because in the carboxylation, in the C4 plants, if you see, one carboxylation is happening here, right? And the second carboxylation will happen in the C3 cycle, right? When CO2 will enter, here the CO2 will bind to RUBP and this C3 cycle will take place. So the carboxylation in the case of C4 is two times. And what about CAM? In CAM also, it will be twice. In CAM also, it will be two times, right? Because here the first time will with the PEP, And when it will enter in the C3 cycle, here it will bind to RUBP, right? So carboxylation will take place twice. Okay. Now, if we talk about the location of C3, C4, and Chem. So in the C3, it happens in the stroma of chloroplast, right? It takes place in the stroma of chloroplast. While in the case of C4 cycle, it is taking place at two different sites. One is the stroma of chloroplast, right? Stroma of chloroplast. And second is, because it has two type of cells, then it also takes place in the stroma of chloroplast of bundle sheath cells. Okay. Or bundle sheath cells. Bundle sheath cells. Okay. While here the location is stroma of chloroplast of mesophyll cells, right? I'm representing it. Okay. Mesophyll cells. So the location here I'm talking is the final carbon dioxide fixation. So it is taking place in the chloroplast of the mesophyll cells, while this is taking place in the chloroplast of the bundle sheath cells. While in the case of CAM cycle, where does it take place? It also takes place. Sorry, it takes place in the stroma of chloroplast of mesophyll cells, right? So here, only in the C4 plant, it takes place in the bundle sheath cells, while in the case of C3 and CAM, it takes place in the stroma of chloroplast of mesophyll cells. Mesophyll cells. Okay. Now, number of cells that are involved. Here, C3, we have single cell, that is mesophyll cell. Here, we have two cells, that is mesophyll and bundle sheath cells. And here, again, we have single cells. Single cells. It takes place during one time, right? While it takes place in two different cells, but at one time. So here we can say that it has spatial differentiation. Spatial differentiation, meaning that the entire dark cycle, it is taking place in or divided into two spaces. One is the bundle sheath cell and one is the mesophyll cells. While in the CAM, it is the only one type of cell, but there is time difference. So it will have temporal difference. The reaction takes place in the temporal differences. Okay. Okay. Now we know about the C4 cells. It has a lot of properties. And yeah. So this is about C3, C4 and CAM cycle differences. Okay. Now, yeah. Okay. So one important thing, if we talk about the primary acceptor of CO2, primary acceptor of CO2 in case of C4 and CAM, what is it? It is PEP, phosphoenol pyruvate. Phosphoenol pyruvate, right? That is PEP. While in case of C3, we know it is RUBP. So this is one of the important comparison, or you can say difference between C3, C4 and CAM cycle. Now, if we talk about the last point, that is about the productivity. So we know that C4 plants, these are most productive. So in terms of productivity, so the most productives are C4 followed by C3 followed by CAM plants. Okay. So with this, we have completed the cycle or the dark reaction in different type of plants. Now, one last thing is about the difference between the Rubisco and PEPcase, because we saw that the Rubisco is used for different purpose while PEPcase for different, but ultimately they are part of the dark reaction. So what is the difference? So if we talk about Rubisco and PEPcase. So the Rubisco, it has both carboxylase and oxygenase property. Both carboxylase and oxygenase property. Okay. While in the case of PEPcase, it has only carboxylase property. Only carboxylase property. Okay. Now, location. If we talk about the location, so in the case of C3 plants, where it is located? In C3 plants, ribisco is located in the mesophyll cells. Mesophyll cells. While in C4 plants, it is located in bundle sheath cells. Bundle sheath cells. Okay. But in the case of peptidase, the location in C3 plant, it is absent. C3 plants, it is, we do not have any peptidase. While in the C4 plants, it is present in the mesophyll cells. Mesophyll cells. Okay. And in the CAM plant, ribisco is present again in the mesophyll cells. While in CAM plant, this peptidase, it is also present in mesophyll cells. Okay. Okay. Now the next is substrate, or you can say, yeah, substrate. So apart from carbon dioxide, we know carbon dioxide is substrate. We have RUBP as substrate for ribisco. While in the case of peptidase, the substrate is carbon dioxide plus PEP, that is phosphoenol pyruvate. Okay. Now, what are the products that are formed by ribisco? So the products that are formed is, the first one is phosphoglycerate or phosphoglyceric acid, right? Phosphoglyceric acid. And this phosphoglyceric acid is the first stable product of C3 cycle, right? It is first stable product of C3 cycle. And in the case of first one is the phosphoglyceric acid. And when phosphoglyceric acid is formed, when ribisco acts as carboxylase. When ribisco acts as carboxylase. Okay. And the second product that is formed is phosphoglycolate. Phosphoglycolate. And this phosphoglycolate, it is formed when ribisco acts as oxygenase. Right? And this is the first stable product of C2 cycle or photorespiration. First stable cycle of first stable product of C2 cycle, or you can say photorespiration. And this phosphoglycolate is 2 carbon, right? Phosphoglyceric acid is 3 carbon. And what is the product that is formed in the case of peptidase? So the first product is oxaloacetic acid. So oxaloacetic acid is the first product that is formed. And this is what? 4 carbon. Okay. So with this, we have completed the dark reaction, or all the 4 type of cycles that are involved in dark reaction. Now the last topic that is left is factors affecting photosynthesis. So that thing we'll cover tomorrow, and we'll see few questions. That's all for today. Thank you.